Hello, it's time to talk about some cellular metabolism. So we're going to talk about what metabolism is and why we care about this in this class, and why we eat, which is one of my favorite things. Let's see. Okay, so let's talk about metabolism. So metabolism is an organism's, it's the sum of all chemical reactions in an organism. You've probably heard the word metabolism before. If someone has, like we all say, well, she just has a quick metabolism. You're talking about someone in general terms who processes energy really fast. Sorry, that's not general terms. You're talking about somebody who can eat whatever they want and they never seem to gain weight. Well, we all have metabolism. We all have a kind of metabolism, and it has a very certain rate to it. So what metabolism is the sum of all the times you form bonds between molecules in your body. Then you do that through a dehydration synthesis and other anabolic reactions. We're going to talk about what those words mean. But you can think about anabolic. I always remember it because anabolic steroids are the steroids that build you up. And so an anabolic reaction is a reaction that builds things. Okay, so that is when you build molecules. When we're talking about metabolism in layman's terms, usually people are talking about breaking things down, maybe, who knows, but in science, we can talk about both the formation of bonds and the breaking of bonds, and the breaking of bonds happens through a reaction called hydrolysis, and the sum of all these reactions are called catabolic reactions. I remember this, so if anabolic is how, I remember this using the steroid example. I always think about my cats break things all the time, so catabolic reactions break things down. It works for me, however you need to remember it is perfect for you. Okay, so let's talk about those two crazy words, dehydration and hydrolysis. So these are just two general examples of very simple chemical reactions that seem scary at first, but a dehydration synthesis reaction is where you have two single molecules, two monomers. They could be polymers, but let's keep it simple. We'll say we have some chemical here, some chemical here. We want to build it to make something bigger, like you're building cell walls. You're building muscles. You're building skin cells. You're building hair particles, whatever. If you want to build this bigger, when these two molecules come together to form a bond, they pop out a water molecule, because one is going to have an H kind of sticking out over here, and one's going to have an OH sticking out over here. And when they come together, you end up with a polymer, and water is also a byproduct. So water leaves, and these two things come together. So this is when you're building things. This is an anabolic reaction. On the other hand, if you're breaking things down, you have some bigger molecule, like you just ate food and you need to break it down in digestion. Part of that happens through hydrolysis, where you have a water molecule that comes in, it attacks these bonds, and by doing so, the water breaks up into two molecules, an H stuck to this and an OH stuck to that, or vice versa, and you end up with two smaller pieces. So you can see that the reason why we drink water, one of the main reasons we drink water, is so that we can power these digestion or hydrolysis reactions. And these are kind of hard to remember which one's which, but I always remember dehydration synthesis is where you are losing water by creating something. So if you're making beef jerky, you're taking the water out. That's a dehydration reaction, I guess, or recipe, I don't know. Whereas a hydrolysis, the word hydro and lysis, that is when water cuts something. So water is coming in and basically karate chopping this molecule into two smaller pieces. Oops, skipped ahead. Okay, so when these things happen, let's talk really quick. So in desert animals, where bears basically almost never get any water, and they're eating food that's really high in oils and not water, because that's how the food lasts longer. If you're talking about seeds or something, they pair these reactions perfectly. So like in a certain desert mouse, it never drinks any water, so it makes sure really carefully that whatever water comes out of this dehydration synthesis is immediately used in this hydrolysis synthesis. So we don't do this because we are around water so often, and we were when we were evolving. So we always have in a healthy human, you always have access to healthy water that you can always drink in and take in new water for your hydrolysis reactions. So we get rid of water that we make through dehydration synthesis, through urination, through breathing, through sweating, and through defecation. So This is also important because we can think about when you're forming bonds or breaking bonds, there's two different forms of energy involved, and energy makes everything go round, and that's really the big take-home message of this lecture is what's happening with energy. So kinetic energy is any kind of thermal energy or heat. It's also considered the heat of motion if you're talking about a physical situation. In biology, when we're talking about kinetic energy, we're usually talking about heat being produced. So when you're digesting a lot of food, you get kind of sleepy because a lot of your blood's moving your digestive system, and everything's getting really hot in your digestive system because you're just breaking down all these bonds. And when you break down bonds, you're releasing energy. When you form bonds, you're storing that energy for later. And so a form of potential energy in chemistry or in physiology is chemical bond energy. And so if you think about if you are storing, if you're a plant and you're storing carbon that you made from carbon dioxide and sunlight and water, you're going to store it in a complex sugar. Well, we then take that sugar and we break it down, and we use that heat that comes from breaking that bond to create our own potential bonds when we're building muscles and tissue and other kinds of things. So the more potential energy you have, the more kinetic energy you can get out of it. And so this relates back to our biomolecules lecture, because the lipids we were looking at had a whole lot more carbon bonds. The more carbon bonds a biomolecule has, the more energy you can get from it, which is why we tend to think of carbohydrates. You can get about 4 calories per gram of carbohydrate, but you can get up to around 9 calories of energy from a lipid, because it just can store more energy because it has more bonds, more carbon bonds. So how do we transform energy? We are these magical, amazing energy transformers. Well, everything's magic until you figure out the science behind it. We actually have figured out really well pretty well, how we absorb energy and release it as heat in our metabolic water products, and we'll talk about what that means. Basically, we absorb energy and we release it as heat if it's not being stored in another chemical bond for other reasons. So in order to think about this, we have to think about these laws of energy transformation or thermodynamics. So energy can be transferred between different forms. So like chemical potential energy can become heat. The heat of a reaction in one place can become a bond energy in another place, but you can never destroy or create energy. It has to just be transformed. And while you're transforming energy, you're always losing a little bit of energy. It doesn't matter how perfect your system is, you're always going to be losing a little bit of energy to something called entropy, which is just the basic energy background of the universe. So every reaction is going to use and lose a little bit of energy. So when let's see, so there's two big kinds of reactions. If you think of all the chemistry in the universe, there's a kind where you have two reactants coming together, you put in a little bit of energy, and then the products are storing a whole bunch of energy for later. So this is like photosynthesis, when you're building up fat stores for later use within your body, that kind of thing. You're taking some reactants that have a certain amount of energy, you store them in bonds like double bonds and triple bonds or something, in carbon bonds that can then be used later. You're basically saving that energy for later. On the other hand, if you're breaking down food or something, you have a lot of energy in high energy food, you start breaking it down, you lose some of that energy to heat. So that's what digestion is really hot, because a lot of energy is lost because we're not very efficient at digesting. But we don't have to be, because we can get more food. We can just keep getting more reactants and get as much product as we want based on that. And then we can meet our energetic needs that way. So I'm kind of alluding to this idea that organisms require energy to live, and we need to think about where that energy comes from. So when we couple those reactions that release energy with reactions that need energy, we can get really efficient systems. So basically, if you are breaking things down in the digestive system, and then you also have some you have some you have the bloodstream going by the digestive system, taking that really hot that heat that's being produced and taking it somewhere else. That heat can then power other reactions just by being present there. You see this a lot in very high energy, very fast swimming fishes in the ocean. They, like marlins and some of the high speed, like the makos, the sharks that are very fast swimmers, they are technically cold-blooded or endothermic or exothermic organisms. They can't control their body heat, except their blood system kind of helps them control their body heat by moving the blood that they need to power their brain and their muscles past the digestive system during digestion, which heats up the blood, which can then go heat up the rest of the muscles and make the fish move faster. So it's kind of a nice way to pair these two energy reactions. So you have this dehydration reaction where you're splitting something, and when you do that you get a little bit of energy, and you can put that so that it's happening right next to a synthesis reaction where hydrolysis reaction where two things are. Oh, I did that backwards. So this is the hydrolysis reaction. So if you have a hydrolysis reaction, you're breaking this bond, you're losing energy. Well, if that energy can then kind of be grabbed by this the dehydration synthesis reaction, if you need energy to be able to create a bond, well, hey, there's energy from this, so we're just going to pop in there and create our molecule. And that is super efficient and fabulous. And look, I stole this from AP Biology slides. Good enough. All right. Okay, so organisms are considered endergonic. So this means that if we don't eat, we die because we run out of energy. So we can make energy, but only if we have energy to start our reaction. We can make our own energy up to a point in that we can start breaking down stuff that we're storing, but we need to take in a whole lot of energy in order to keep things going. So why do we even need energy? We are always building biomolecules to keep our body going. Our body is always going, well, not always, but reproduction is an incredibly costly process energy-wise. It takes a whole lot of energy for reproduction to happen. This can be if you're really, really stressed or if you're malnourished, you can skip periods because your body just cannot use that energy, cannot waste, quote unquote, waste that energy making reproductive things if it can barely keep itself alive. So reproduction takes a whole lot of energy. We can feel that movement can take a lot of energy. If you've been working out a lot and you haven't eaten in a while, your muscles really start to feel weak. We haven't gotten to, well, we'll talk a little bit about active transport later, but anytime you're moving chemicals against a gradient, so basically against their natural flow, you have to input energy into that system or it's just not going to do it. I'm not going to do it if you don't basically pay me to do it. And we also need to regulate our temperature really carefully because our chemical reactions that make up the whole of our body only work within a certain range of temperatures. So we have to make sure we regulate these things very carefully. And all of these things are reasons why we need energy, so why we have to eat. So this kind of goes back to the idea of energy coupling. We use those catabolic reactions where we break things down and release energy in digestion to fuel our anabolic reactions, our synthesis reactions, where we're putting two things together, but we need energy to make it happen. So that pairing is a nice, neat way to keep things running smoothly. So let's see. So how do we fuel? So some people like to make this analogy that in order to run our body, we need some sort of currency. It's like if you're going to run the country, you need money, or you need money that people trust. And so in this case, we have an energy molecule that's very high. It's very high. It's a high kind of currency. It has a lot of energy in it. So when we eat high energy organic molecules, these things like carbohydrates, lipids, proteins, nucleic acids, like we were saying, we break them down through catabolism, there we go. And then we need to capture that energy. So how do we actually capture that energy? You can't, as soon as it becomes heat, it's lost. It just diffuses out of the environment. So what can we do to trap it in some kind of bond before it is lost to heat? And so that's where this idea of an energy currency molecule comes in. And we found it, we know it works really well. And it's a way to pass energy around from places where catabolism is happening, digestion is happening, to places where anabolism needs to happen, but you need energy in order to do it. And what we use is a molecule called ATP. And this is a nucleic acid. So let's go on. So ATP stands for adenosine triphosphate, and it's this modified nucleic acid, or modified nucleotide. So it's adenosine is used in DNA and RNA, but this very specific flavor of it, if you have adenine, which is the nucleic side, or sorry, this side, the nucleic acid nucleoside, and then you attach it to this ribose. So this is the RNA version, not the DNA version, because it's got two oxygens here. It's not deoxy, it's just ribose. And then you have if you only have one phosphate, it's called AMP. And this is a really negative molecule, this phosphate group, because we've got 3 oxygens. They're all very negative in nature. Phosphate is a very polar group, so it's got a kind of negative aura, if you want to think of it that way. It has this negative cloud around it. And then suddenly you want to shove in another phosphate and then another phosphate. Well, these two bonds, since you're forcing these very negative things together very closely, these two bonds, you have to put a lot of energy in them to make them stick. And so by putting a lot of energy in them, that means when you break them down, you get a lot of energy back. And so you can kind of marvel at this efficiency of biological systems, because we can build this very simple energy carrier, and we can break it down when we need energy. We can basically just pop this off, and then we can rebuild it when we have energy that we need to store for later. So that's a nice simple system. So every time you add one of those phosphate groups, it gets harder and harder to add. So when it's just one phosphate group, it's called AMP for adenosine monophosphate. And then we add another one, and then we add another one, and then at this point, this is a very unstable molecule. And in the case, if this were a nuclear reaction, this would be very dangerous, because when you release nuclear energy, you get a nuclear explosion, and that's a lot of energy. But in the case of this bond energy, it's just enough energy to make things work in your body, and so it's perfect. So once you get to the 3-phosphate stage, this becomes a very high energy molecule, and you have so much stored energy in each bond. The most energy is in that last phosphate, and it's also the one that's hardest to keep it bonded to the molecule, which might be a really bad idea, except the point of it is you're going somewhere else in the cell to make something happen, or somewhere else in the body to make something happen. If you need that energy to happen really quickly, you need it to be able to pop off really quickly so you can access it immediately and not have to wait for it to come unbonded. And so it's kind of a spring-loaded system that lets it just pop off and release its energy right when you need it. So it becomes a very excellent energy donor, as we see there. So what does it do? It basically, as soon as this pops off, it releases a bunch of energy, so about 7.3 kilojoules of energy. And if that energy is nearby something else that needs to be made, they can just kind of absorb that energy in and use it in their bond production. So this is called phosphorylation. This is when a released phosphate just literally moves to another molecule, and it destabilizes that molecule. So then when you phosphorylate it, it causes it to do something. So we'll talk about that a lot. Anything that is phosphorylated, or any enzyme that phosphorylates something or makes something happen, is called a kinase. So if we see ASE, that's an enzyme. It means it's a protein catalyst that makes stuff happen faster and decreases activation energy. An example of this was the cytokines we saw in the last lecture, where we were talking about cell differentiation. So their longer form name is the cytokinases. So they are enzymes that cause a cell to determine what kind of cell it's going to be. So you're going to be a blood cell, you're going to be a white blood cell, you're going to be a red blood cell. It's a cytokine that makes that happen by phosphorylating certain parts of the DNA to make them active. It's more complicated than that, but that's a simple way to think about it. So once we have energy, what is the point? Why do we even need to do it? Well, we need to make proteins so that we can make stuff happen in the cell. So remember that all the information for making every protein in the body is stored in our DNA, but we don't access our DNA just directly. That's like, if you access the original copy of something over and over and over, you're gonna break it down. If you break it down, it could get misread and weird stuff could happen. So the DNA is same in every cell in the body, except for the red blood cells, where you actually lose the nucleus altogether. We'll talk about that later. Remember, DNA stays in the nucleus and is double-stranded. It has those A, C, G, T nucleotides, so the A and T bond together, C and G bond together, and it's that order of bonding that creates the genes that then can be read in a certain expression as proteins. So when we're bonding these things, the CG bond is stronger than the AT bond, because there's 3 hydrogen bonds happening instead of 2. So if you have a region of the DNA you really need to access, you fill it with a bunch of AT bonds. The CG bonds are a lot harder to access, so that means these are genes that are only active in certain parts of your life or under certain characteristics or under certain environmental stimuli. So these are going to be, anything that's really rich in AT bonds is going to be something that's activated a whole lot, whereas things that are very rich in CG bonds are going to be activated much less frequently. So since DNA is double-stranded and it's kind of like it points in one direction this way and then it points in another direction this way, that means that you can use one strand as a template to make the other, which is nice. So when you're doing DNA synthesis, you just read down one and you read down or up the other in kind of little fits and starts, and you end up getting two brand new DNA copies off of two DNA strands. So it works perfectly. Well, not perfectly, but it works as good as we want it to. So if you were to look at just one gene and what is happening, so let's say this codes for an adorable dimple or something, and you just look at this is supposed to represent, this is you, this is your essence and your being, and it's a bunch of squiggly colorful dots or lines. What does this mean? So what this is telling you is there's something else happening. So we have this combination of A, T, C's, and G's, like here's a whole bunch of T's in a row, here's a whole bunch of A's in a row. What does it mean? Why does it matter? Well, we have to then take them and move them onto something else, and so they can be read in a different way. So the genome is all the genes all together in our entire in all of your chromosomes, and all 46 of your chromosomes. We can kind of generically refer to those all as the genome, and it's kind of like this is kind of like a book that has everything in it. So that's all the DNA in the person. Once you have within the genome, you have individual chromosomes, and so that's kind of like the chapters in a book. And these are there's 46 segments of let's see, there's 46 basically sections to the DNA that get condensed in a certain way, and there's 23 pairs of them. Within the chromosomes, you have different genes. So if we're still following our book analogy, if the genome is a book and the chromosomes are the chapters of that book, within the chromosome within that chapter are sentences. So genes are little chunks of specific chromosomes being read. We have probably 25,000 genes that we know of. That number may even have changed as of like yesterday, because we're always figuring out more and more things. Things that we thought were individual genes are turning out to be the same, and then things that we thought were quote-unquote junk DNA are actually incredibly important, so it's a very fluid number. The codons are where within the gene you make three little triplets, or you make triplets of 3 nucleotides, which are kind of like reading a word. Like if you look at this word, you know it's the word, but you only know that because you're getting these letters in a very specific way, right? So if you have the word gene, but it's spelled E-N-E-G, well, that's not the word gene anymore. That's something totally different. So the order of the codons is very important, the same way the order of the letters in a word are really important. And what these do is in groups of 3, they code for an amino acid, and we're going to talk about that. And within each codon, it's if we're following our book analogy, if a gene is a sentence made of codons or words, then nucleotides make up those letters. So if this is a gene, let's say this one side of this is a gene, I have so that would be a sentence basically. I can go and I can count in 3s, and every 3 is a codon. So 3 letters of the DNA that codes for an amino acid. But each one of those codons is only made of 3 nucleotides. All right, so here's the problem with making with making proteins. How do you turn this process of making proteins on and off easily? And how do we basically take all the information stored in the DNA and turn it into something functional that's not just a moldy old book sitting in a closet? It is a very functional and useful productive part of society, for carrying that analogy out even more. How do you there are 20 different amino acids. How do you create 20 different amino acids from only 4 different letters? And it comes down to, so this is called transcription. So this is where we're taking the RNA and we're turning it into protein or amino acid chains. And when this happens, so first we have so let's go back one. So transcription is when you take DNA and then you take a little chunk of it that you want to read or have active and you turn that into RNA, okay? So when that's happening, you have this one protein, this RNA polymerase that sits on the DNA and connects the nucleotides to start making RNA so that an RNA can then start feeding out of the cell once it's done. RNA is a single-stranded and unstable molecule, so it doesn't last very long, which is good because if you have a signal for like histamines to fight a response, you don't want to be activating that all the time. You want it only for as long as you need it. Whereas DNA is made up of A, C, T, and G molecules, RNA gets rid of the T and uses uracil instead. Apparently, it's just more stable that way. And again, this happens through the protein RNA polymerase. So once transcription has happened and you have that RNA out in the cell, so it leaves the nucleus and it's now in the cytoplasm, you can start translating it. So this is where you unlock the code of the RNA and you turn it into working amino acid sequences, which can then become proteins and then carry out your genes. So what happens is that RNA is going to slide into a ribosome like we talked about last time, and as it's sliding through the ribosome, translation is happening. So one ribosome strand can actually have a whole bunch of different little polypeptides that look like little macaroons to me, or sorry, different ribosomes sitting on them. And as those little ribosomes are scooting along, they're producing amino acid chains. And the way they do that is they read in groups of 3. So they read these codons, and they say, okay, so A, C, C. Then what is that going to make? So that's the DNA. Then the codon that's made in the RNA is going to be what matches it. So A matches with U, C matches with G, da, 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 da. So then these 3 things are going to code, we've figured out, they only code for tryptophan. Okay, so cool. So we're going to go in groups of 3. These are going to make tryptophan. These are going to be read by the ribosome as something else, as this phenylalanine, maybe? I don't know. And then these 3 will read for something else, etc., etc. And so if you have a triplet code, so if only one nucleotide codes for one amino acid, you can only have 4 amino acids. If you have a doublet code where you have 2 nucleic acids coding for an amino acid, you can only have 16 different amino acids. But a triplet code where you have 3 different things can give you 64 different possible combinations. Well, it turns out we only need 20 because there are only 20 amino acids. But here is basically the secret code of all living things. So this is how we know life evolved from one thing, because this is a very simple and yet complicated system, and everything, all the living organisms on the planet and even some non-living organisms use this. So this code is used to translate information from the RNA into proteins, and so this always starts with a sequence of RNA that's codes for AUG, and it starts with this methionine or met amino acid. Then let's say the next RNA sequence is CGU, okay? So you'd say, how would I read that? I'd go to this chart and I'd say, okay, my first base is C, so that's here, and then my second base is G, so I'm going to go over CG, and then U, oh, there it is, so whatever that was, it coded for arginine, the amino acid arginine. But this is important to note that if it's off by a single one, you suddenly change your strand, okay? You change your potential RNA, your potential protein coming out of that RNA. So that a typo in the DNA means a change in the RNA sequence, which could then change the protein sequence, maybe. So if GAG gets misread as GAA, so let's go back and read. If I have GAG, which is GAG, it gets read as GAA, it's actually going to code for the same thing. And this is called wobble, where that last amino acid isn't as important as the first 2. But you'll notice if you misread GAA or GAG as GAU, you get a different one, and that's where mutations come up. So this could be the difference between blonde hair and brown hair, or it could be the difference between a functional and a non-functional protein in the hemoglobin in your blood cells. So it can be, most mutations are incredibly, most mutations are benign, but a lot of mutations are dangerous, can actually hurt people or hurt an organism. The chance of getting a good functional mutation is incredibly rare. Okay, so what does, this is so many stuff, this is so much stuff, what does the work in each step? Hint, important to know. Um, in the transcription step, we have an enzyme called RNA polymerase. In the translation step, there's an enzyme built into the ribosome itself. And actual translation, oh, and part of that ribosome is also being done, or sorry, that ribosomal enzyme activity is being done by actually a different flavor of RNA, which is called transfer RNA. So this is a lot of information, and why does it matter what's happening? Why is it important? It's really important that we control our genome to make sure we can control our bodies through something called homeostasis. Basically, if your body starts to get too hot, you need to be able to control how you're regulating temperature to decrease that body temperature, or you're going to start malfunctioning. So that control of all bodily functions or bodily systems is called homeostasis. So we're going to talk about that. So why does the body do a certain thing? Probably to maintain homeostasis. So why do you secrete a certain hormone at a certain time? Probably to maintain homeostasis. So maybe things are a little out of whack, and you have to move them back to being within normal parameters, basically. So our genes can be regulated to help control homeostasis. They can be regulated to help us conserve energy. So if you are doing something and you're really hot and you're using a lot of energy, you start sweating to kind of deal with that excess heat. Again, and it's kind of like regulating temperature, but again, gene regulation helps us control energy usage and production. And it's incredibly important to control our cell specialization, because if you have unchecked cell growth, or if you're creating the wrong kind of cell in a certain place, you end up with cancer. And that, as we know, can be a deadly proposition. So how do these genes get controlled? So we can first control genes by controlling the production of the protein itself. So we can say, no, how about we don't read any DNA today? We don't need to make any of whatever that DNA wants us to do. We can control the destruction of a protein. So if we want to prolong a signal for longer, we just don't break down the protein, because the protein is what carries out the signal. So we just let it keep going for as long as possible. We can control the production or destruction of RNA, because remember, it's the RNA that takes the DNA and translates it into a usable protein. We can do negative feedback inhibition, which is where if something is happening, so let's say you're producing a whole lot of testosterone, and so this is done through a process that starts in the brain. It takes in all these sensory signals. It goes to these signals are summed up in the hypothalamus, which releases a certain hormone, which then tells the pituitary, hey, we should start releasing testosterone. And the pituitary says, got it, we're on it. And the pituitary sends a signal to the testes and says, it's time for testosterone. Or in the case of a secondary place to get testosterone is the adrenal glands, including in women. So basically, these signals go to those glands, and they say, "Oh my God, it's time for testosterone." But if that didn't then get shut off, that signal, you would never stop making testosterone. So as soon as testosterone is in the bloodstream, some of it will start to head back up to the brain, and as soon as it hits the brain turns itself off with the signal that it had originally set out to make. And so that is negative feedback, or an inhibition pathway. There are examples of positive feedback, such as contractions during birth. You have a hormone being produced called oxytocin. When it hits the bloodstream, it goes back to the brain and tells the brain, hey, there's oxytocin, let's make some more, so we can start doing contractions and push this baby out of us. We're going to get into these more examples of this in so much more detail. There's so many other reasons to regulate your genes and how to regulate your genes, but this is a nice overview of very basic cellular cellular metabolism. So it's important to know the difference between dehydration reactions and hydrolysis reactions, and anabolic and catabolic reactions. And very important to remember that it's the ATP molecule that is the energy currency molecule of the body. Very important, very high energy if you have 3 phosphates on it. And then we went into how proteins are made very simply through translation, and the importance of why we have to regulate our genes through protein regulation and signals feedback and everything. Have a good day, and let me know if you have any questions. Bye.